is another diagram of the cell cycle, your restriction points. Okay, so you have G1 to S, the restriction point, right? If a cell does not have enough signals to progress through G1 and then begin to synthesize DNA, it will exit the cell cycle and enter something called G0. Right? G0 is like a holding pattern. The cell will still do its job, skin cell, liver cell, whatever it might be, but it's not actively dividing. Give me an example of a cell that you might find in G0. In your normal body, doing normal function, but it's locked in G0 forever. A nerve cell. Why? A neuron. Let's say a neuron. Why? So neurons don't necessarily reproduce. Okay? What other cells don't reproduce very quickly? Or very much? Cat? What'd you say? You said something. Or breath. Muscle cells. Okay? Muscles. Skeletal muscle cells. Right? Cardiac muscle cells. They don't necessarily reproduce quickly. They live most of your life. Neurons and muscle cells are found in G0. Okay? And then, when you get a green light or a go-ahead signal at G1, the cell will progress through, begin to synthesize DNA, and then begin to go through the cell cycle. And then you have your other checkpoints. G2 checkpoint, and then the mitosis, chromosome attachment to microtubules in mitosis is your mitotic or M checkpoint. Okay? Questions on that? All right, so there are signals from the outside that help progress or tell the cell to continue or stop dividing. Okay? So one of those outside signals are growth factors. Growth factors are proteins or hormones, stuff that is going to, molecules that will tell the cell or force the cell to increase those positive regulators. All right? Will keep the foot on the gas, per se, through the cell cycle. Okay? So you have, this is a little example of a growth factor experiment. Okay? And actually this is done pretty often. All right? So a sample of human connective tissue is cut up into small pieces. Let's say, anyone have an ACL surgery ever? Let's say, in a way, we could take your damaged ACL, that cartilage tissue, take it out, cut it up, put it in a plate. Those cells are there. They haven't been necessarily growing. Okay, they're kind of stuck in G0, but they're living and maintaining your what was normal ligament. Okay? You have a ligament tear, we'll take out those pieces, we'll put them in a petri dish. And now we'll add a growth factor. Okay? Those cells are not, they're outside of their normal environment, they're going to start to grow. Okay? And now we add a little bit of a growth factor. All right? This we could get without a certain growth factor or with a certain growth factor. We add the growth factor and those cells begin to grow quicker. Right? That growth factor, and this is called one example called platelet-derived growth factor. There's tons of growth factors out there. There's insulin-like growth factor. There's epidermal growth factor, blah, 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 blah. Okay? But outside of the normal environment, they're exposed to more signals that are going to do what? What's in the presence of this growth factor outside of the normal microenvironment where your cartilage cells are, what are they exposed to? Those cells are exposed to what? Not plate. Well, let's just say a growth factor. Okay? And the environmental signal is going to say to the signals to those cells, there's space to grow. we need more cells. So the cells begin to grow. Now we grow those cells up, let's say, in a certain form, okay, and we remove them from this environment, and we can put them back into your knee to repair your ACL. That is the basis for what we call tissue engineering, okay? And this has been done for growing ears on mice. Human ears on mice. Okay? The way they've done it, they've taken cartilage cells out of the human, put them on a protein scaffold. What is a scaffold? A support system. Okay? So we take these cartilage cells out of the human, put them on a support system in the mouse. The mouse is going to produce all the signals, the growth factors that are necessary. The cells grow on that support system in the shape of an ear. Then you take it off and you put it on, you transplant it back onto a human. That is actually the type of science that is ongoing in tissue engineering right now. So we could grow cartilage, replacements for ligaments. They're looking at replacements for cervical discs. They're looking at replacements for heart valves, aorta, blood vessels, you name it. Okay? Questions on growth factors. But it all comes down to taking the cells out of their normal environment and then increasing the signals, increasing the inputs that make them grow, and then you control their growth in a certain way, and you can do these fun types. All right? Now, most eukaryotic cells have signals that will regulate their growth and stop them from growing too much. Okay? Most eukaryotic cells are requiring a contact. They're what's called anchorage dependent. All right? So what you see here, all right, they require a surface for division. They contact to something, whether it be other cells, a plastic surface, something. They need to be, most cells of the human body don't just grow floating around. Okay, they're usually attached to something. And when they are, that's what's called anchorage dependent. Okay? And they also exhibit something else in culture called density dependent inhibition. Right? So they'll grow out to a certain point, and then they run out of space. Right? So here you see all the cells are occupying all the available space. They're touching another cell. Most of the cells in your body are touching another cell, right? So what does that tell those cells? Well, hey, I'm here. Don't push me out of the way, right? Don't overgrow this space. You have your personal space. I have my personal space. At that point, most normal cells of the body stop growing, all right? So they have to be contact to something in order to grow. But then when they run into other stuff, boundaries, they stop growing, right? So we have anchorage dependence growth. They have to be touching something in order to grow. And we have density dependent inhibition. They run into each other, right? They become too dense. Okay? So this is an example showing here, this is what's called a scratch wound assay. You take a full plate of cells and you remove some of them. What do? You clear some space. What are those cells that now have no crosstalk between them? There's no boundary to another cell. What do they start to do? They start to grow. Why? Because there's space to be filled. Right? They would what's called assimilate the space. Right? You grow into your surroundings. All right. Now, the difference comes when we talk about cancer cells. Okay? Cancer cells are going through the cell cycle uncontrollably. Right? They remove those checkpoints so they continue to grow. those checkpoints are also removed. Those other things that control their growth are also removed. So they don't necessarily have to be anchorage dependent anymore. All right, so that when they leave the tumor, say that originated in the breast and eventually found they get into the lymph node and eventually go to the lungs, that they've removed that attachment point. They moved from one place to another. That's the basis for metastasis, or the movement of from one tumor place to another. All right? They do not, cancer cells no longer have density-dependent inhibition. They'll start to grow over and on top of other cells. Right? That's what's called invasiveness of a tumor cell. All right? So cancer cells, unregulated cell cycle progression, and then there's no more anchorage dependence for growth, and no more density-dependent inhibition. They remove all that stuff. Questions on those? Okay, so now here's sort of an idea of the progression of a tumor. Okay, so here's a primary tumor, the breast cancer tumor. In this case, it's a non-ductal carcinoma. All right. Eventually, a tumor grows from one single cell at its point. A single cell that is going to divide faster than the cells around it. Okay, so then it goes from 1 to 2 to 4 to 8, and it's going faster and faster every time it divides. That forms a tumor. Okay? At this point, it's a localized tumor, okay? But that tumor then loses its anchorage dependency. At first, it loses its density-dependent inhibition. Right? Most of the normal cells in the breast or in the local area are not growing because they are attached to other cells. Right? They're getting that input. That tumor cell loses that property and starts to form a tumor itself. It continues to divide, even though there's lots of cells there. Doesn't matter. Okay? And then it begins to invade other tissues. That's the invasiveness. Okay? At some point, the tumor loses its anchorage dependency. It separates from the tumor itself or invades the nearby blood vessels or lymph nodes, and then will start to move throughout the body. Okay? That's when you end up with metastasis. And it ends up going to the lungs, the nearby lymph nodes, other sites away from the primary tumor site. Questions on anchorage-dependent growth and density-dependent inhibition? Cell cycle progression and how it relates to cancer itself. Okay, so I wanted to show you this. This is sort of a way, experimentally, how we measure cells in the cell cycle. Okay, so we can take cells out of a dish, and we can do things. Okay, we can treat them. Let's say we have cancer cells in a dish, and we're going to treat them with a new experimental chemotherapy. Okay, we have to be able to see whether or not that chemotherapy works, right? So how are we going to measure that? If our idea is we're going to treat this cancer cell with this new drug that's going to cause them to no longer be in the cell cycle or no longer progress through the cell cycle in a faster fashion. We want them to be held where? In G1, right? So one way we can measure that is the amount of DNA that's present in the cell at that time. How much DNA is present in the cell in G1? All right, we'll say 46 chromosomes. Okay? During S phase, how much DNA is present? Up to double that, right? Up to double that. At G2, we know that DNA synthesis has finished. So we know that definitely G2 has how much DNA? 92, double the normal amount. Okay? So if we could somehow measure the amount of DNA, we could separate where the cells were in the cell cycle. And that's what you see here. Okay? By taking a dye that goes and intercalates or tags DNA, dyes DNA, and fluoresces under a certain light. Okay? We can measure the fluorescence of a cell based upon its DNA content. And then we can count those number of cells. That's what's actually happening here. So in this A part, A represents the number of cells, okay, at a certain fluorescence. And you'll note that, say, we have 200 versus 400. What is that? What's the factor between those two? 2. Okay, so if these number of cells have 200 amount of fluorescence, and these cells have 400, how much relative DNA do they have? Those in C have how much more than those in A? Double. What does that tell you? Which ones of which peak are found in G1, which peak is found in G2? 200 G1. The normal amount of DNA has a certain amount of fluorescence. Double the amount of DNA has double the amount of fluorescence. Okay? What's that area between A and C? S phase. Why? What's ongoing in S phase? Synthesizing. But the cell isn't necessarily completed with it. So it can have anywhere between the normal amount and double the amount and be in S phase. Okay? This is called flow cytometry. All right? It's a way of signaling where cells are in the cell cycle. Okay? And you can see this is showing treated versus untreated. So what is the treatment, our chemotherapy, doing? Jason?
Increasing the number of cells in G1.
Increasing the number of cells in G1, meaning they're no longer dividing. Meaning that the restriction point is working. Meaning that our drug is stopping cells from progressing through the cell cycle. Okay? Questions on that? Okay.